Microfluidic droplet arrays for high-throughput screening of catalytic reactions are revolutionizing the way scientists discover and optimize chemical reactions. Have you ever wondered how new medicines, materials, or energy-efficient chemical processes are developed? Traditional methods for testing chemical reactions require large amounts of reagents, expensive equipment, and a lot of time. But what if we could shrink the entire process into tiny droplets, each acting as an independent mini laboratory? This is exactly what microfluidic droplet arrays achieve. They enable scientists to run thousands of chemical reactions in parallel, using only microscopic amounts of materials, making research faster, cheaper, and more efficient. Imagine you are a scientist trying to discover the perfect catalyst, a substance that speeds up a chemical reaction. Instead of running one reaction at a time in large flasks, microfluidic droplet arrays allow you to create thousands of tiny reaction droplets, each testing a slightly different condition. These droplets, only a few nanoliters in size, can be precisely controlled and monitored, allowing researchers to rapidly identify the most effective catalyst with the highest yield. For example, in drug discovery, pharmaceutical companies use this technology to test how different enzymes speed up reactions for drug synthesis, leading to the development of more efficient and environmentally friendly drug production methods. But the impact of this technology goes beyond pharmaceuticals. In the field of sustainable energy, microfluidic screening is being used to develop catalysts that improve fuel cells, hydrogen production, and carbon capture technologies. Instead of relying on trial and error, researchers can systematically test thousands of catalyst compositions and conditions in just a fraction of the time it would take using conventional methods. Have you ever considered how much time and resources are wasted in large-scale chemical experiments? With microfluidic droplet arrays, scientists can significantly cut costs and environmental waste while accelerating discoveries. Another fascinating example is in artificial photosynthesis, where scientists aim to mimic the way plants convert sunlight into energy. To make this process efficient, researchers need to test countless combinations of photocatalysts, materials that absorb sunlight and trigger chemical reactions. With microfluidic droplet arrays, they can rapidly screen thousands of photocatalysts at once, identifying the best candidates for clean energy production. Could this technology change the way we approach chemistry entirely? Instead of working with large test tubes and slow processes, microfluidics brings chemistry into the world of miniaturization and automation, much like how computers transform data processing. In the future, we might see AI-powered microfluidic systems that not only conduct thousands of reactions simultaneously, but also analyze and optimize them in real time. This could revolutionize industries ranging from medicine to energy, creating more sustainable and efficient solutions. What other areas of science and technology could benefit from high-throughput screening with microfluidic droplet arrays? As automation, miniaturization, and AI-driven research continue to evolve, the future of chemistry may no longer be in large-scale laboratories, but inside tiny, precise droplets. Unlocking discoveries at an unprecedented speed, microfluidic droplet arrays enable the compartmentalization of tiny reaction volumes, allowing high-throughput screening of catalytic reactions. These systems use precisely controlled microfluidic channels to generate, manipulate, and analyze microdroplets containing reactants, catalysts, and solvents. Unlike bulk phase screening methods, microfluidic droplet arrays offer precise control over reaction conditions, reducing reagent consumption and enabling thousands of reactions to be tested simultaneously. This technique is particularly useful in optimizing catalytic performance, identifying novel catalysts, and accelerating reaction kinetics studies. A microfluidic droplet array was used to screen enzyme-catalyzed reactions in synthetic biology, improving the efficiency of biocatalysts used in pharmaceutical production. Microfluidic droplet arrays function like a miniature chemistry lab, performing thousands of individual experiments in tiny liquid compartments simultaneously. Homogeneous catalysts, which operate in the same phase as their reactants, are widely used in fine chemical synthesis and pharmaceutical production. Microfluidic droplet arrays allow researchers to screen different catalyst formulations by encapsulating them in tiny droplets, ensuring rapid mixing and precise reaction control. This method reduces diffusion limitations and enhances reaction efficiency, making it ideal for screening transition metal catalysts, organocatalysts, and enzymatic systems. Microfluidic screening of palladium-based catalysts has enabled the rapid discovery of new reaction conditions for cross-coupling reactions in drug development. This process is like speed dating for catalysts, rapidly testing multiple candidates to find the best match for a specific reaction. Heterogeneous catalysts, such as nanoparticles or solid-supported materials, often suffer from mass transfer limitations in bulk phase reactions. Microfluidic droplet arrays provide a controlled microenvironment where catalysts are evenly dispersed, maximizing their surface area and improving reaction kinetics. Additionally, the ability to tune droplet size and composition enables precise control over reaction pathways, facilitating the optimization of catalytic performance for energy and environmental applications. Microfluidic arrays have been used to evaluate metal oxide nanoparticles for photocatalytic water splitting, advancing sustainable hydrogen production. These systems work like an espresso machine, ensuring that reactants and catalysts interact efficiently under controlled conditions for maximum output. Enzyme-based catalysis plays a critical role in biomanufacturing, food processing, and pharmaceutical synthesis. Microfluidic droplet systems provide an ideal platform for screening enzyme activity under varying conditions, such as pH, temperature, and substrate concentration. By encapsulating enzymes in individual droplets, researchers can monitor their catalytic efficiency in real time. Microfluidic screening of lipase enzymes has improved biodiesel production efficiency by identifying optimal reaction parameters for fatty acid conversion. This system functions like a chef testing different cooking techniques in separate mini kitchens, finding the perfect conditions for the best results. Electrocatalysis is fundamental to energy storage and conversion technologies such as fuel cells and batteries. Microfluidic droplet arrays enable the rapid evaluation of electrocatalysis by generating controlled droplets that act as microreactors for electrochemical measurements. This approach facilitates the screening of metal catalysts, electrode coatings, and electrolyte compositions, accelerating the discovery of high-performance materials for renewable energy applications. A microfluidic electrocatalysis screening platform has been used to identify cobalt-based materials for efficient oxygen evolution reactions in water electrolysis. This method is like testing different battery designs in miniature circuits, determining the best combination for maximum energy efficiency. Multi-depomented catalytic systems, such as bimetallic nanoparticles or hybrid organic-inorganic catalysts, require extensive screening to determine optimal compositions. Microfluidic droplet arrays enable combinatorial testing of catalyst formulations, where different metal ratios and ligand structures are tested in parallel. This approach significantly accelerates catalyst discovery in complex reaction networks. A droplet-based combinatorial screening approach identified an iron-nickel alloy as an efficient catalyst for CO2 reduction, supporting sustainable carbon capture efforts. This process works like mixing paint colors to find the perfect shade, exploring countless combinations to achieve the best catalytic performance. Microfluidic droplet arrays allow real-time monitoring of reaction kinetics by tracking color changes, fluorescence, or electrochemical signals within individual droplets. This enables researchers to study reaction intermediates, activation energies, and transition states at an unprecedented level of detail. Such insights are crucial for designing more efficient catalytic processes and understanding fundamental reaction mechanisms. Microfluidic single-droplet analysis has been used to study the kinetics of enzymatic oxidation reactions in biofuel cells, optimizing energy conversion efficiency. This technique functions like slow Mac play replay in sports, capturing every detail of a reaction to understand its underlying mechanisms. Artificial intelligence AI is being integrated with microfluidic droplet screening to autonomously analyze reaction outcomes and optimize catalyst performance. Machine learning algorithms process high-throughput data from droplet reactions, identifying trends and suggesting new experimental conditions in real time. This self-optimizing system dramatically reduces the time required for catalyst discovery and process development. An AI-enhanced microfluidic platform, optimized enzyme catalyzed carbon fixation, improving the efficiency of artificial photosynthesis systems. This system is like an automated recipe tester, adjusting ingredients and conditions dynamically to create the perfect reaction. As industries shift toward greener and more sustainable chemical processes, microfluidic droplet arrays are emerging as key tools for optimizing catalytic reactions with minimal waste. Future developments may integrate renewable catalysts, bio-inspired reaction pathways, and continuous flow microreactors to enhance process scalability. These advancements will be crucial for pharmaceutical synthesis, environmental remediation, and sustainable energy production. Microfluidic screening of biodegradable catalysts has led to eco-friendly plastic production methods, reducing reliance on petroleum-based materials. This approach is like designing a smart factory, minimizing waste and maximizing efficiency in chemical production.